This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 1089, recorded on February 16th, 2024. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Fort Lee, New Jersey, Dixon Despommier.
Hello, Vincent, and everybody else. Let's see, a brief overview of the weather. Looks can be deceiving is the name of today's weather. It's beautiful out. The sun is shining. There's hardly a cloud in the sky. And if you were to go outside without a coat, you may not come back because it's below freezing. It's like 28 degrees and wind, lots of wind. So a very, very deceptive looking day.
What is it Daniel quoted yesterday about the weather? This is really good. Let me get it here. It's right here in front of me. No, it's not. He said it's something like, the problem with the weather report is it's right often enough so you can't ignore it, and it's wrong often enough so you can't rely on it.
100% right. Absolutely. Now that's accurate.
It's 6 degrees C and mostly sunny here in New York City. Also joining us from Western Massachusetts, Alan Dove.
Good to be here. It's 38 Fahrenheit, 3 Celsius here, and also blustery. Wind northwest at 22, gusting 29 miles per hour. So it's
challenging landing a small plane in that kind of.
I would not. No.
You just keep flying until you.
No, I just would not take off into that in the first place. There's a very easy solution for this.
I see. Okay.
And from Madison, New Jersey, Brianne Barker.
Hi. It's rather similar here, 41 Fahrenheit, 5 Celsius, and rather sunny. We had snow for the first time in a while earlier this week, so we actually have snow on the ground. And I think we're supposed to get at least a little teeny bit more this weekend.
We had snow on Tuesday night, I think it was earlier this week. And it was forecast to be 9 to 13 inches, and then the evening forecast changed that to 2 to 3 inches.
Exactly.
But in the meantime, they had already canceled school.
Right.
So.
Yep. The AI can't even help us here.
No.
AI. If you enjoy these science programs, what is it that Nels Ld said? People are saying such good things lately on these programs. Let me pull up the latest TWiV. He said we are building spaces that celebrate science.
I
know.
like that. I do like that.
I don't know. I guess if we built an incubator, but
yeah, you built a space that celebrates science. You really did.
That's right. That's right.
I guess so, but not spaces, right?
He means virtual spaces.
Well, knowledge doesn't exist in a vacuum, so you have to.
If you like that, if you enjoy building spaces that celebrate science and hearing us talk about science, we'd love your support. Microbe.tv/contribute. It doesn't have to be huge. It can be a few dollars a month, but we'd like it, and we'd like it every month because we use it for our expenses, which grow on a monthly basis because a nonprofit is not simple to run. You have to keep track of everything. I don't remember anything past last week, which means I have to have other people doing it. Anyway, microbe.tv/contribute. Viruses of Microbes 2024 is going to be in Cairns, Australia. Cairns, Friday. Let's see, what's the date? It's this summer. Oh my gosh. I should have this, right at the tip of my tongues. But there's a website, vom2024.org, 15th through the 19th of July. So I'll be going to do a TWiV, and Jolene is attending the meeting, so she's going to join me on the TWiV, and we'll have a couple of guests for the meeting.
Now, that's the middle of winter
I was
in
just
Australia,
about to
but it's
point
in
that out.
North Queensland, so it should still be warm.
Oh, no, it's warm at that time of the year all year.
Yeah.
It is a tropical paradise, basically.
That sounds fabulous. I feel like I want to go to the meeting just because it sounds
You
fabulous.
should do it. You should do it.
You could go.
No, really, you should do it.
You have to fly. I guess
A
you
long
fly to
time.
Sydney and then you fly up to Cairns.
No, you don't. You can fly to Hawaii first and winter over there for a while, and then continue on a
Well, I'm
couple
just
of
saying
weeks
there
later.
there are probably no international flights to Cairns.
Or you can catch the trade winds from San Diego west to Fiji and then down to no.
Cairns is not too far away from Brisbane, so you could fly to Brisbane direct and then get a puddle jumper out to Cairns.
Cairns.
Sorry, Cairns.
Drop the I and the R.
Right, Cairns.
So anyway, check that out. It is vom2024.org. And also don't forget the Rosenfeld Lab at the Food and Drug Administration. In particular, the Center for Biologics Evaluation and Review. She has a research assistant position where you could go in the lab and do experiments on enteroviruses. You could investigate cross-reactive antibodies and their roles. You can help make animal models for enteroviruses. And all this is BSL-2 work, so you don't have to suit up. And if you want to know more, we'll have a link to a PDF in the show notes, and you can email Amy Rosenfeld. And don't forget to buy The New City, which is Dixon's new book. It is receiving accolades.
I
Accolades.
think someone bought it.
There you go.
Yeah,
She bought it.
my copy's in the living room. Yeah.
We all have
My
one.
20 copies are in my closet. Could I make a plea at this moment? The book is doing well, but not great. And it's probably because of the lack of the number of reviews. So if anybody has bought the book already and hasn't written a review, I would really encourage you to do so.
It's easy.
And it's easy. And Island wrote a wonderful, very long review. And you don't have to write one that long, but just an honest opinion about what you thought of the book. And I would very much appreciate that.
Can do.
Great. Thank you.
Also this week, Paul Offit published a book. It's in the other room. It's something like, please tell me when it's over, something like that, about the pandemic, obviously. And we will have Paul on in March to talk about the book. Okay, The New City. Now, no, this is not right. Yes, it is right. Some news bits. We have a bunch of people who sent us articles about the first known fatal case of Alaskapox. Randall, for example, wrote, "You might be interested in this article in our local paper about the first known fatal case of Alaskapox. The virus was first identified in 2015, but all the earlier cases were mild. The individual was immunocompromised." And he sent an article in the Anchorage Daily News. State reports first known fatal case of Alaskapox. And then there's also an article in the Alaska Beacon.
I don't know if it's been named yet, but I think a new group of viruses should be part of that in the cryoviridae.
Because it's so cold.
It sounds right.
It's cold, right? Yes.
Well, in the summer it gets warm.
Mm-hmm.
Yeah, unfortunately, that is true.
I asked Rich, what is this? He said, I know a little bit. Discovered 2015, hangs out in rodents, voles, I think. Interesting in having its own branch inside pox phylogeny, but closest to cowpox, though branched off at the very root of cowpox, scattered zoonoses, so in this respect behaves like cowpox. I don't know symptoms in humans, but if I had to guess, it would be like cowpox. Guy who died was old and a transplant patient, in any case immunosuppressed. So, interesting phylogeny, but otherwise no big deal. I think it's interesting that it's named after the state, and I can't think of any other virus that is. Do you guys?
No, not at all. And I'm also just surprised that it was named after the state in the first place.
Right.
Yeah.
And
The 2015
normally, I
is-
mean, back when we were naming them after places, it was usually a more specific thing. You know, even maybe you could get away with this in Rhode Island, but Alaskapox, that's kind of a vast area to.
Yeah.
Yeah.
Well, you have viruses in the Flavidae group.
It should be Fairbanks pox.
Fairbanks pox. That's right.
So you
Or-
remember in 1993 they tried to name Sin Nombre Muerto Canyon virus,
Mhm.
and they said nope.
Nope.
Yeah, but
So-
St. Louis encephalitis exists. That's a
Yeah,
Well,
that's an
right.
old
If you go back 20, 30 years further, then people were not so upset about having Norwalk virus named after the town of Norwalk in what state, Dixon?
I believe it's Ohio.
Ohio, yes.
Just a wild guess. Just a wild guess.
So the other one is Coxsackie virus, right?
Yes, that's right.
1947. People in Coxsackie
right.
are proud to have a virus. I have someone on the live stream who lives in Coxsackie, and she thinks it's really cool.
Yeah.
Semliki forest virus. Don't forget that one.
Yeah, yeah.
That's in Russia, I believe.
Yeah. Do you know the story of Semliki forest virus?
I don't.
So there was a guy in Africa. He had isolated it, and he asked someone, "What's the name of the place where we are?" And the guy said, "It's Semliki." And so the guy called it Semliki Forest, but Semliki means forest because the guy said we're in the forest,
I
Right.
see.
whatever language that is. You
Right.
guys know that story?
Stop to ask for directions.
didn't-
Where
I did
am
not.
I? You're in a car. Yeah.
You're in a coma.
very, very helpful guide there.
Is
Well,
there a
Ebola
virus?
virus.
Yeah, is there something with California in the name? I'm sitting here going through states in my head trying
Yeah,
to think.
California encephalitis, Western equine encephalitis is, but it doesn't say exactly where. Though.
Hmm.
Yeah,
But
I'm
the
thinking.
Ebola certainly says where it
There's
is,
Japanese encephalitis virus.
Yeah.
Right? That's a
That's a
broadly
whole
based.
country.
That's a country. That's right.
I mean, there's VSV Indiana.
There was the Spanish flu.
I'm just like I said, I'm going through states in my head.
Yeah.
That doesn't
Right,
get passed.
right,
But when you
right.
talk about that, most people only hear VSV, right?
Right.
Yeah.
The Indiana is a lab thing. There's a VSV New Jersey, I
That's
believe.
true.
Oh, really?
Yeah.
Did not know
So
that.
once you get past the name, but anyway, I couldn't think. If anyone can think of one, let us know. Okay.
Anyway, so
that's,
Alaskapox, generally mild, sporadic cases, zoonotic, and this fellow. You said transplant patient, but some of the news reports say he was being treated for cancer. In any
Okay.
case, he was immunosuppressed
It was
and got a bad
immunosuppressed, yeah.
fatal infection. So try and stay away from the voles in Alaska.
Indeed.
Well, what did I hear? Oh, Banco Schmidt said Wednesday night 30% of rodents west of the Mississippi have the
Hanta.
bacterium that causes plague.
Plague.
Oh, yeah.
That's right. That's right. That's
Mhm.
right.
This
That is
is
right. Pasteurella pestis,
which-
it used to be
no,
called.
it's not Pasteurella pestis.
Ursina.
Yersinia.
Yersinia
Yersinia.
pestis. You were going to
Yeah.
say Yersinia, huh?
Well,
Yes, there's sort of a fabulous historical story behind why that is, and there are great books about it. So if that's something someone ever wants to geek out about history-wise,
We have an article from Emerging Infectious Diseases where they have looked. This is very interesting. So MERS coronavirus is endemic in dromedary camels from the Arabian Peninsula and Africa. Over 75% seroprevalence. And, eventually, now and then it gets into people. So people always want to know how that happened. So in this study, they did they sampled workers in an abattoir in northern Kenya. An abattoir, of course, is a slaughterhouse, and there they slaughter camels because people eat them. And they sampled camels. Oh, they sampled camels, not the workers. I'm sorry. And they found it was a biphasic pattern of seroprevalence in the camels, 2 times in the year. I think they also looked in people because the title says biphasic MERS coronavirus incidents in nomadic dromedaries with putative transmission to humans in Kenya. So these are nomadic dromedaries. So they have some evidence of transmission to slaughterhouse workers because some of them were positive as well.
So why isn't there a big MERS outbreak like there was in the beginning?
Well, there wasn't really a big one in the beginning.
That's pretty dramatic.
It was a
though.
single case. There was a series of cases in South Korea where a guy acquired the infection in
Yes.
Arabian Peninsula. He flew to Korea. He had a respiratory disease. They stuck him in the hospital. They didn't know about MERS yet, and they didn't do any infection control, so he infected a bunch of other people.
Yeah, and I think part of it also was that he went to multiple hospitals to try and get a diagnosis, and so he was in multiple hospital waiting rooms.
Yes.
So yeah, I think that it doesn't transmit very well person to person,
Yeah.
unless it's in one of those healthcare associated situations. And so that's, I think, the reason why the outbreaks are particularly small with this, is that we're not seeing a lot of person to person. You have to come in contact with a camel.
Yeah.
Never becomes a SARS-CoV-2. Never becomes a human virus. Okay, so
So
in this
far.
paper they have 7 out of 48 abattoir workers who were seropositive. And none of
so
them
the
have had
biphasic pattern that they see in the camels, right, peaks in October and February, which I think that correlates with transitions between wet and dry seasons. Is that right?
Yeah, that's correct.
Sounds right.
And they looked at by ELISA, and that's where they find the antibodies. But none of these workers had any symptoms, as far as they could remember. All right. Then there's an interesting article in CEPI News. CEPI is a Center Coalition for Epidemic Preparedness and Innovation. It's a nonprofit that raises money and then tests vaccines, pays for vaccine development. In this case, they're paying for rapid on-the-spot diagnostics for Nipah and Lassa viruses, because we need that, and nobody seems to want to do it. So CEPI is pumping some money into it, and hopefully something will come out of that, because we need it. We need to know who's got one or the other.
Yeah, and you would want to have tests that could be done in the field in the places where people get these viruses.
Exactly. This is peripherally a comment peripheral to what we were just discussing, but I had a wonderful Zoom conference with 2 students from Indiana University who wanted more information about building a vertical farm, and their whole idea was to use the vertical farm to prevent Nipah virus in Africa by moving whatever farm there was outdoors indoors and keeping out the bats. And they had a wonderful course in epidemiology. I was very impressed with the 4 students who were part of that small group. I said, you need to be a bigger group, because you have a terrific idea, and it needs to be heard by people that can do something about it.
Our next little news item is from CDC. Human papillomavirus vaccination coverage in children 9 to 17 years of age in the U.S. in 2022. 38.6% of children 9 to 17 had 1 or more HPV doses. It's not a lot, folks. Not a lot. Children with private health insurance were more likely. The coverage was 41.5%. compared to children with Medicaid coverage, 37%, other government coverage, 30%, and those without insurance, 20%. It's expensive. So that's why you see this. This is not good, because this prevents cancer in both men and women. So you should vaccinate your kids. And the government should pay for it. The government should just pay for everything. Oh, no, not everything, but healthcare for sure. And I'm working on education, too.
Yeah.
But that'll happen.
Everything.
I think there are 2 things that every human on Earth should have. And you can add your own, but these are my 2. These are Vincent's 2. Education and healthcare.
Yeah.
Nobody should go without either.
I would agree.
It's not fair.
I would add shelter, clean drinking water.
Well, yeah, that's
things.
Good too. I agree with that, but I can't ask for everything. You have to add
food
it.
would be good to include on that list. But Those programs largely exist in a lot of parts of the world, and what Vincent is saying is programs that don't exist in all countries, even
Right.
maybe very wealthy countries that don't provide those things to everybody.
That's right. My big wish is that everybody is literate. And once you can read and write and make judgments based on what you read, then the rest will follow.
I think that falls
That's
with
usually
Vincent's
true.
education.
Yep, that
Usually,
falls under
yeah,
education.
absolutely,
It's
absolutely.
the
I think
first
that's a
R.
great idea.
Yep.
It's a good idea. All right, and finally, our last little bit here. Hey, guess what new infections there are this week? Yeah, in the U.S., measles. More measles. Arizona
Jeez.
and Minnesota.
You'd think it was infectious.
Well, you know what, Dixon? Vaccination will prevent you from getting the disease known as measles.
Is that right?
And I do not understand why. I guess now it may also be access, right? Maybe a lot of parents don't have access,
Yeah,
but the government ought to pay for it.
The government does pay for a lot of these vaccines, and this is one. I mean, childhood vaccination is pretty widely available, and I think a lot of the failure to vaccinate is not related to availability.
Right.
Yeah,
Well,
I also wonder in the HPV case how much of it has to do with the age group. I think kids go to the pediatrician with more regularity when they're a little younger, and
Yeah.
may get some of the vaccines, the younger vaccines, more frequently than maybe they do when they're slightly older.
Right.
But all of these kids should be getting these vaccines.
There's also a well-documented effect, I've heard it called the Kodak effect, where the second child often doesn't get vaccinated as promptly as the first child. Kind of like you have more pictures of your first child. Apparently, I only have one. So, but yeah, the parents, yeah, we did this already. We're not gonna. We'll get them vaccinated when we
Cell
get
phone
to
effect.
I think it's more that they're so busy with two kids.
That too could have an effect.
All right, onwards to the literature. Our snippet today, PNAS, virus-derived circular RNAs populate hepatitis C virus-infected cells by Shan Kao, Pak Boom Boon Shuen, Shu Shen Shen, Xiaoha Lei, Kunlaya Sumbun Wiwat, and Peter Sarno. Peter Sarno I know very well.
Yeah.
He followed me in the Baltimore lab as a postdoc. I went out the door the day he came in. So it's about circular RNAs, very interesting molecules, and most eukaryotic cells have them. They are made by a weird splicing mechanism. So splicing, you take a precursor to mRNA in a eukaryotic nucleus, and introns, which are sequences in between the parts that code for the protein, they're removed by a process called splicing to form the final mRNA. But sometimes the splicing can go in an opposite direction, and you get circular RNAs as a consequence. And eukaryotic cells can have like 25,000 of these circular RNAs per cell. And
this is one of those things that was discovered after I got out of graduate school. And, I was fully aware of what the basics of molecular biology. And then along came this finding that there's all this, well, first of all, that the circular RNAs get produced, and then that they're everywhere, and they're apparently doing things, and we have some clues about what some of them are doing, but no idea what the rest of them are doing. And there's this whole additional level of probably regulation that's going on throughout eukaryotic cells that just popped up.
And I think that it's important to think about them, because I also don't think about them particularly often, but I think we're probably underestimating them because they're really stable and they can soak up other stuff. So they can bind to and maybe sequester or use, or who knows what, other proteins, other nucleic acids, things like that. So if you start to imagine that, you're suddenly like, whoa, there's a lot of stuff that could be going on and regulated by this.
It's kind of like that dream where you discover all these extra rooms in your house. It's like, whoa, where'd this come from? Yeah.
Discovering Malcolm Malkovich.
Yes.
So yeah, they can act as sponges, as Brienne said, to hold other things. They actually can bind proteins that bind RNA, or they can bind microRNAs.
They don't
And
actually, though, transcribe anything, or they do?
translate?
Yep. Mhm.
Well, we're going to see in this paper that they
Ah.
do, or they can, yeah. But they're mostly made from DNA viruses, right? Because DNA viruses undergo splicing of their mRNAs. And
Right. So on the viral side, they mostly come from DNA viruses. On the cellular side, they're just coming from mRNAs, right?
Mm.
Yeah.
yeah,
Oh,
they're coming
yeah.
from
Although in this
mRNAs.
paper, they're coming from an RNA virus.
Yes. So right, we're getting we're getting
Sorry.
a little out of sequence here, but
Yeah,
Okay,
no,
we're
no,
finished.
That's
right.
the
So these are
question. They wanted
these are
to
things that
know-
exist in cells that are produced in mRNA splicing that are doing all kinds of probably important stuff.
Yeah.
It turns out viruses, DNA viruses, have been shown to have some of their transcripts spliced into these little circles. And then
Right.
we get to
Yeah,
this
but-
paper.
We want to know if they're in RNA viruses. So they looked at hepatitis C virus infected cells, and that's what this paper is about. And they said they looked at circular RNA libraries, and they say, astonishingly, we found hundreds of putative, they call them VCirc RNA species from the 10,000 base hepatitis C virus genome. So the HCV genome is a long, positive, single-stranded RNA, which is packaged into the enveloped virus, which is it's a hepacivirus.
Right. I was just
and-
about to do Rich's little introduction.
Yeah, you can. Go ahead.
No, that's all right. You just did it.
It's
it's,
good enough.
Long
and-
single-stranded RNA that
and at the 5' end of this RNA is an internal ribosome entry site. So these RNAs are not translated by the usual method of the ribosome interacting at the very 5' end with the cap structure, but they internally initiate and then translate the plus-stranded genome. And so they found lots of small circular RNAs. They're most of them from the positive strand. They have a couple of clusters. They're not random. They're not all over the place. They're from certain places. One place near the 5' end, and the internal ribosome entry site was intact in some of those. And then they had a couple of other clusters, but they range in size from 130 nucleotides to 2,000 nucleotides, with an average of 200 to 300. So this is a bioinformatic interrogation, because they're looking at libraries, and they wanted to verify that this actually happens in virus-infected cells. So they extract RNAs and they reverse transcribe the RNAs into cDNAs. And what they do is they know where these circles are coming from the genome, so they can make primers that will bridge the circle at the junction. And those places in the genome go in opposite directions. So if there are no circles made, you would never see them by amplification.
Can you make
Right.
interfering RNAs to that process?
Yeah, we'll get to that.
You know,
You got the right experiment.
I
It's good.
don't have the full article because I can't pay that much
Oh,
for this particular.
Didn't you get the email? Is that what they say? Didn't you get the email?
Probably
There's a-
not.
Vincent put
No,
the
I
papers
didn't
in.
get the message. I did not get the message.
It
Get
was
it?
a message. You're right. I
Yeah.
sent a message, not an email. I'm
It was a
putting all the
faulty
papers.
translate.
I'm putting all the papers in a folder in the Google Docs from now on.
Ah,
So you
that's-
no longer
okay.
have an
okay.
excuse to not read the papers.
I don't.
But
I
you
was
do
so-
ask the right questions, and that's fine. All right, so they do PCR to amplify these, and they find that in fact they do see these circles. They have verified 14 predicted circular RNAs by this kind of PCR, so that's good. They do exist in virus-infected cells. Then they ask, are they translated? Now, for years, a lot of people didn't believe that there was this non-canonical translation method where it involves an internal ribosome entry site. They said, no, it's an artifact, blah, blah, blah. It's got to bind to the 5' end. The ribosome has to bind the 5' end. So Peter Sarno did an experiment where he circularized an mRNA and showed it would not be translated, which is true for the 5' end model, unless you put an internal ribosome entry into the circular RNA, and then it would be translated, proving that you don't need an end to translate as long as you have an iris. There's some clever thing there. I can just see it forming.
It probably is, yeah.
So they decided to see if these circular RNAs could be translated with, some of them have an iris in them, remember, as I mentioned, they're derived from the 5' end. And so they put some GFP, green fluorescent protein, into these circular RNAs, and in fact, they can be translated. And they look at it in several different ways, by green fluorescence and also by luminescence as well, bioluminescence. So these RNAs can be translated. Let's see, where is that? I guess it's later. Okay, so what about functions?
Well, they
Dixon,
do also have an siRNA for Dixon here.
Yes.
Against
Dixon,
the junction site.
So they used an interfering RNA to attack these, basically.
Yeah, they make small interfering RNAs to decrease the abundance of specific ones.
I keep forgetting how much I've remembered.
Yeah, in infected cells. And they show that when you knock down the cluster 1 or 2, which are 2 abundant clusters of circular RNAs, you reduce the abundance of viral RNA in infected cells, and you reduce the yield of infectious virus. Figure 5C. Let's see.
So stop there.
They
What
actually
effect
measure viruses by infectivity.
All right. When you get rid of all that extra viral circular RNA, then what happens to the activity of the cell that it's found in? Does it do something different and better, or is it neutral?
What do you mean? Like, does it not kill the cell? They don't actually say that. No, they don't report it.
Well,
It's an
yeah,
interesting
but
question.
it does say though that lots of the peptides produced are pro-viral function.
Yeah.
The word pro-viral, when you get rid of it, does that make it a pro-cell function?
Well, when you get rid of it, there's less virus, and I think less virus would be pro-cell.
More
But
metabolic activity
they didn't
towards the
look
cell.
in greater detail at the. I mean, they'd already found a pretty darn cool thing.
Oh, absolutely. Well, it could get even cooler.
Yeah.
They need at least something for the next paper.
Yes.
So this is an interesting slight divergence. So the replication of hepatitis C virus requires 2 cellular microRNAs to bind to the 5' end of the genome, and they're liver specific. So the virus does not reproduce in other tissues because they don't have these 2 small RNAs which seem to stabilize the 5' end. So a company has made antagomeres. So MIR is a microRNA, and they've made antagomeres that bind to this miR-122 and prevent it from binding the viral RNA. And it's an antiviral, basically. And it actually works, except the other ones that have been developed are much better, so nobody uses it.
The other question that would arise at that point then is, what are they doing there to begin with without the virus?
Well, they have a cellular function, actually. They have a role in cholesterol metabolism, yes.
So I'm going to ask then, if you interfere with that process of a normal cell activity, then what happens?
The patients who went through the trial were fine. It's a transient treatment. It's not forever.
Okay.
So, but the point of this is that you can make antagonists of RNAs. And so in theory, you could antagonize some of these circular RNAs as an antiviral approach if you wanted. But it's not ever happening because the double and triple therapy that we have, the direct acting antivirals, right, which act on viral polymerase and other proteins, those are great.
Yeah, but I don't think finding more HCV antivirals is really what they're going for here. This is a phenomenon that if HCV is doing it, I would bet that many other RNA viruses are doing it too.
Mm-hmm.
And we already know DNA viruses are doing it. I think this is going to be a pretty widespread phenomenon.
What you're saying, Alan, is if we don't look, we won't find.
That's exactly right.
That's the deal.
That's very wise.
Yeah.
And one of the weird things, I mean, there are a few weird things going on here, but one of them is HCV replicates it's entirely in the cytoplasm, right? And RNA splicing is in the nucleus. And they don't, I think they looked and they don't see any evidence that the splicing enzymes are getting pulled out of the nucleus to do this, or that the virus is going into the nucleus to do this. So how
up?
is it getting these circular RNAs made?
Right.
Yeah, it's a good question.
That's a great question. Yeah, I was more on the wait a minute, if the virus is making these and these are so stable and perhaps more stable than RNA, what does that do in terms of, infection in the cell and sort of persistence in the organism and things like that? And if these are acting as sponges, could they be impacting a lot of other cellular processes by attracting other cellular proteins or things like that? Those were the places I went off of it.
Yeah.
So outside of the cell, if you had a collection of these RNA molecules that had not circularized yet, could you just shake the test tube and all of a sudden they're all circles? Could they self-anneal?
Well, we don't know the mechanism by which they form, right? Alan was just
Well, that's what
saying,
Alan
so
was asking. And if they
we don't
could
know.
self-anneal, then you wouldn't need to
It's
go
more than
anywhere.
that. It's a covalent bond that's formed, right, to make
Oh,
a
wow.
circle.
Yeah, I mean, you need the actual back splicing.
Yes,
So you'd think you would need the splicing factors.
but they're
And
not in the
yet-
cytoplasm.
and yet-
Right.
I think
Splicing
some
factors.
cytoplasmic extract biochemistry is in somebody's
Yeah.
future.
There you go.
Absolutely.
There you go.
Absolutely. And by the way, there are some peptides made from these little circles, and so they actually
Yeah, that's what I was
overproduce the peptides in infected cell, and it increases viral expression, gene expression. So they're proviral, and that's really interesting to see what they're doing. I'm sure they're looking at that now.
Proviral
I'm-
meaning
asking. Not pro-life.
helping the
Yeah.
virus, not
Helping.
proviral in the
Yeah,
retroviral sense.
that's unfortunate.
Little
Yeah.
confusing bit of terminology there.
I wouldn't have used proviral. Yeah.
No.
But every time they voted viral.
Yes. These are not amateur viruses.
They are not amateur viruses by any means.
Anyway,
And they live by a
that's-
circular economy.
This is introducing many people to circular RNAs, I'm sure,
Yeah.
who don't know about. But I talk a little bit about them in my lecture on transcription and RNA processing. What's really interesting is that cancers seem to have a circular RNA signature. Right? So they're different from what you would find in a non-transformed cell. And so people are going a little bit crazy trying to figure out if they could use that in some way, right? Because you can imagine anything that's different about a cancer cell is potentially
Absolutely.
useful.
Absolutely.
Right. So that's the
You bet.
value there. Okay, on to some immunology now.
Aha.
This is very cool because, as you know, we're big fans of T cells here, and this is about T cells and a specific kind of T cell. This is an article in Nature, Prevention of Respiratory Virus Transmission by Resident Memory CD8 positive T cells. Do we have co-first authors here? I don't know. Oh, yes, we have. These authors contributed equally. Ida Udabach and Sarah Micheletta, two co-first authors. And then we have Jacob Kohlmeyer, who is the corresponding author. By the way, last week we talked about, remember the measles paper in rodents? Dixon is the only one of you guys that was there. But basically, the two senior authors wrote me and said, "Thank you so much. People told me now they heard it on TWiV, and then they went to read the paper. A new paradigm.
They got a
in
TWiV bump.
literature."
Well, I'm glad they went to read the paper and didn't just rely on TWiV.
That's right. That's right.
Yep. So, we love T cells, as you know, but the vaccines that have been made are mainly making antibody responses that we know of that prevent disease.
Yeah,
But,
I was going to say the vaccines that we think about, we've mainly measured the antibody responses. And
That's
so
right.
we assume that the antibody responses are doing all the work.
Right. And when viruses change, as influenza viruses and SARS-CoV-2 viruses, coronaviruses change, that makes it difficult for the antibodies to do their thing. But T cells, there's not very little change in the virus, and so T cells can always work.
T cells are not easily fooled.
Exactly.
T cells are not easily fooled. That would be a good title
That's
also
good.
for this paper.
There
for
you
this
go.
TWiV.
I'm sorry, Alan. I didn't mean that. It just popped
No,
out
that's
of my
fine. That's fine. That's a good one.
It popped out of my amygdala.
So during the pandemic, we talked a lot on TWiV on how every time a variant emerged and was a bit immune evasive with respect to antibodies, the T cell epitopes were unchanged. And Alessandro Sette came on a few times and talked about that. You can check that because you can make the peptides and you can show that they're not changed in the variant and they can still stimulate T cells. So in this paper, we are looking at CD8 positive tissue resident memory T cells in the respiratory tract, TRMs. T standing for T cell, and then RM is the resident memory T cell. So it's not an effector, it's a memory T cell that could be there, it could hang out there, could have coffee, chat, caffey clatch, all that. And then when bad things happen, they can start proliferating. And
It's that old T cell sitting in the corner just watching things who jumps up
Yeah,
all of a sudden and says,
It's
I know who this is.
it's quiet. It's really too quiet out there.
I remember you from 2019. Yeah.
Picture a Western bar and it's
Yes.
smoky and there's
Yes.
somebody sitting in the corner just with a hat down. And then someone walks in and wants to make a mess and that guy in that or gal in the hat
Mhm.
gets up and says,
Says,
"What are you
so
doing?"
it's you again.
So that's what we're talking about here. And when you get infected or vaccinated, these tissue resident memory cells go throughout the respiratory tract, the nasal cavity, the trachea, the airways, the lung parenchyma, and they're important in limiting replication and immunopathology. They can prevent the spread of infection throughout the lung. And they say here, averting severe disease that can result from viral pneumonia. You know, it's interesting. When you get influenza, it's mainly focused in the trachea, and many people have substernal pain for a couple of weeks, which is actually destruction of the respiratory epithelium by CD8 T cells, cytotoxic T cells. So there's not zero cost of having these lovely T cells.
That's innocent bystander effect.
Yeah, they take a scorched
absolutely.
earth policy once they get going.
Yeah.
Gotcha.
And one piece to sort of mention about this is that in the past, when we thought about memory T cells, we usually thought about them as being maybe present in the blood or maybe going back and living in the bone marrow
Right.
after they've been activated and differentiated into memory cells, with the idea that they would find the microbe again if the microbe came around the lymph node. But they wouldn't be specialized to the tissue of interest where you were first infected. And
Right.
some of the idea was that you needed some cytokines from the bone marrow to keep those cells alive as memory cells. And more often, we're realizing that there are memory cells that are not just in the bone marrow, not circulating around the blood, but that are staying in the original tissue that was infected. And so questions about, how they stay as memory cells and persist are really interesting. But that also becomes really important because we as immunologists often, at least in the past, like when I was doing some of this kind of stuff in grad school, would do our measurements on things like the blood and the spleen.
Mhm.
And the only way you're going to measure a resident memory T cell that is in the respiratory tract is to actually sample the respiratory tract, because these cells are not circulating. They're just staying resident to the respiratory tract.
Right.
And so only when we've started to measure immune responses and figure out ways to actually look at different tissues have we started to appreciate the understanding of these. They would be particularly important because you might imagine if you got SARS-CoV-2, you don't want to wait around for a T cell to come out of the bone marrow and go to the lymph node and find the tissue and then go to the lung. That takes a bunch of work. Whereas if you had a memory cell that was already in the lung, residing there, being able to be that person in the smoky bar that we've talked about, that, the person lives in the smoky bar. It's not like you had to call them up and tell them to come.
Right.
Can be really important, but we hadn't been looking for them for a really long time. So their biology in some cases can be a little bit behind our understanding of kind of the bulk cells. There also may not be as many of them, or it might be really hard to get them. So people have a harder time studying them than
Right.
sort of general memory cells that you might find in the blood.
Do you
And
think
so
they have? Go ahead.
These limitations that Brienne is pointing out are all reasons why it's been difficult to assess the ability of T cells to have an effect on reducing viral loads, preventing transmission. Even another one, which Brienne mentioned, so this is called destructive sampling. Right, if you got to kill the animal to take the T cells or the virus to measure virus loads, that's no good. And Brienne says, now we have ways to measure the cells. You can measure virus too. Without killing the animal, you can make a fluorescent virus or a virus with luciferase, and then you can image the animals. Right?
See, that one they had when I was a grad student, because I've done that one. But
well, you were only a grad student last year,
I wish.
You're so young.
I have a question about why the T cells would remain in the tissue that they were generated in. And are you of the opinion, it's an opinion now, that they have receptors on their surface which match that tissue only? And they lock onto the cells as a result.
The idea is less that maybe that they are locked in that way, but that they have things like chemokine receptors and other trafficking molecules that make them sort of do best or always end up back in the
they stay there
organ.
because no place else feels like home.
Yes, sure.
Well,
exactly.
If they came from the bone marrow, then for sure they're out of their element.
Right.
So that's
right.
Migrated through the thymus and then
this is
stuck in the lungs or something.
Well, no. So the idea is that sometimes maybe after they got stuck in the lungs, originally we thought then they would go back to the bone marrow just because the bone marrow was a nice place to hang out.
Well,
they wouldn't have to go back through the thymus again. They're hanging out with like the young developing cells back in the bone marrow.
Yeah, there's an energy cost to that. And if every T cell variety of every tissue did that, I think that would not make sense biologically. I think this makes much more sense.
Sure. Yes. But then the question is, how many T cells do you keep in the lung before the lung stops being good for breathing and is just T cells?
As we learned on TWiV, the lung is an important organ.
See, I say the important
things. T cells offer a breath of fresh air, basically. Yeah. These are for future shows, not this.
So, and these are T cells that have been generated in response to an infection.
Right.
So, and then after that, they hang out in a tissue where they presumably were left over.
Exactly.
Right?
So, how many?
Also makes sense because it's most likely that you're going to see that same microbe in that same anatomic location. Place. It's probably not going to show up in a completely different anatomic location.
In that vein of still pursuing the biology of this, how many viruses do you know that are absolutely tissue specific and not generalists?
That's a Vincent question.
It is.
I can. Well, yeah, there are both, right? There are generalists, and then there are ones with very specific tropisms. And I would say the generalists are more rare, right? SARS-CoV-2, smallpox, but poliovirus, quite specific, intestinal tract and CNS, rabies virus.
We were just talking about Hep C.
Hepatitis.
Hep C, liver, influenza virus, human influenza A viruses, lung, respiratory tract, right? Epithelial cells.
Sure.
So
should we start looking for those T cells in those places for intractable diseases like AIDS?
So that's a good question. And I would say to Brienne, let me modify it slightly. Are there any tissues without tissue-resident CD8 positive T cells?
I don't know of any tissues without resident memory CD8 positive T cells. If people have actually done the work of extracting them. Different tissue. So I know that there's some really interesting data on resident immune cells, including resident memory T cells in the GI tract and also in the respiratory tract or in the the genital uterine. You know what I mean.
Urogenital tract.
That's the one.
The UG tract.
Yes. But it's, again, you generally are going to have to do things like using a protease to disrupt the extracellular matrix and do a lot of work to get the cells out from tissue layers, which can be, really challenging. So I don't know of an organ where we have not found them though they may be harder to work with and harder to really experiment on in some organs.
Well, can't you light them up in sections without having to remove them?
I think that you probably can. I haven't done it myself, but I think that you probably can.
There must be some markers, A for the T cell and B for the tissue of the T cell.
Well, I think you'd have to do that based on just what tissue you have, and do you find T cells there?
Right, and then do they stay or do they?
Well,
will you stay or will you go?
But see, if you have a tissue, you've done destructive sampling, so you can't then check again later to see if the T cell is still in that location.
You're into a Heisenberg virology system.
Exactly. I think that based on your question about generalists and specialists, this is probably why the immune system has evolved both central memory and resident memory T cells.
Right, because
to cover both bases.
My list of eukaryotic parasites does have a tissue specificity to them in a lot of cases, not in all of the cases, but in a lot of cases they do. And you could then use them as your experimental model for T cell residence or not. And I think you'd probably do very well at the NIH applying for a grant on that subject, because we want to know.
Right. Let's do some experiments, Dixon.
You got it.
All right. So the question here, can we see the CD8 TRM T cells, can they affect transmission? And so they use, as a model, they use Sendai virus infection of laboratory mice. So that's a para-influenza virus that is a natural virus of mice. It infects mice in the wild. It was discovered in Sendai, Japan. Hey, talk about naming a virus after a locale. There you go. And
remember, Sendai got trashed by
it did?
The
tsunami?
The tsunami, yes, indeed. It did.
So the virus is transmitted through aerosols and direct contact among mice. The mice don't get very sick, and so they can use that as a model for transmission. It's been studied quite extensively in all kinds of ways. So,
Vincent, sorry, I was a little bit confused about, and then you sort of mentioned it, so I wanted to be clear. So they're not actually using Sendai here in the mice. They're using influenza that has Sendai epitopes. They talk a lot about PR8.
Right.
And so it's influenza with the Sendai epitope. It's not full on Sendai.
Yeah. I know this is most of the experiments are using that. I'm wondering if there's some later experiments that actually use Sendai. So I don't know why they're making this statement in the introduction that Sendai is a great virus that's transmitted, blah, blah, blah. Let's go through the experiments and see if there are any with actual Sendai virus.
Sendai virus is a great model, so we used something else.
So we have, so what they want to do is...
That confused me.
What they want to do is generate T resident memory cells in the respiratory tract. So they take an immunodominant T cell epitope from Sendai virus, it's from the nuclear protein, and they put it into influenza virus. Right.
Yeah, but maybe they do that because Sendai virus immunosuppresses. And I remember outbreaks of Sendai virus in animal colonies, and it trashed the entire mouse colony, and you had to start all over. So maybe they don't do that because they want to keep the immune system intact.
Anyway, they take this immunodominant T cell epitope from Sendai virus nuclear protein, they put it into PR8. So that's now PR8 Sen-NP. And then they give this to mice intranasally, and they infect with influenza virus intranasally, and they get effector memory T cells, TEM, in the circulation, and then they get T resident memory cells into the respiratory tract. And what's cool is that they do not get Sendai-specific CD4 T cells or Sendai-specific antibodies. So you
that might be why they do the recombinant.
Yeah. They don't want to get it confused.
Yeah, but if they infected with Sendai, you get everything, right?
Yeah, exactly.
However, they do talk about using Sendai as a great transmission model.
Right, exactly.
It is, but still.
But it's not in this paper. Okay, so so that if you do that intranasally, you get nice TEMs in the circulation and TRMs in the respiratory tract. But if you do intraperitoneal inoculation of influenza virus carrying the NP of the peptide, you get similar TEM in the circulation, but less TRM in the lung and nasal cavity. So that's actually experimentally going to be useful for them.
It also
Okay.
makes some sense.
But do you
The
get
T cells are not going to preferentially want to go to the lung if they did not first see the antigen in the lung.
So do they go back to the peritoneum?
They need an elementary education.
Yes.
Oh my gosh, so many arcs here. Okay, so this works in a way that they want it to work. So then they're going to do their challenge experiment. So here we go. Here's the Sendai part. They immunize with this PR8NP, and then they challenge 35 days later with Sendai virus encoding luciferase.
Right.
And then they use that luciferase as a measure of viral load, and then you could see it transmitted to other animals. So that's
So your vaccine is not Sendai, but your infection is Sendai, and Sendai is good because it transmits easily between the mice, and then you can look at transmission dynamics, and they put a luciferase in there so you can get these
there you go.
glowing pictures that are printed really, really tiny.
They're so small.
It Sendai a big message, though, to the immune system.
Yeah.
All right. And so they use bioluminescence as a surrogate for virus titer because they say there's a good correlation. And that makes it easier. It makes them not have to kill the mice, right? And so they can do these long-term experiments, right? So mice immunized with PR8-Sen-NP had the greatest reduction in viral load in the nasal lavage samples, and they had a reduction in the duration of infection. So shorter number of days that they're actually shedding. If you immunize intraperitoneally, so that's immunization intranasally with the T cell peptide. If you immunize intraperitoneally, you get a more modest reduction of viral load, and you get a more modest reduction of duration of infection. Because as we said, the cells are not, you don't make as many TRMs in the lung when you do that. That's
and so that kind of implies that the TRMs are the ones that are responsible for that clearance and that initial protection, because this only is seen in the immunization type that gives you good TRMs.
It would be interesting, though, to see if instead of being in the respiratory tract, they were in the peritoneal cavity, just waiting for the viral. Come on, where the hell is this stuff? And it's never going to come because it's
totally
the wrong
true.
place.
So
that's a switcheroo.
So they want to know now if there's any impact on transmission, because none of these immunization protocols actually completely eliminates virus. It's not sterilizing Sendai virus, right? So, but maybe it would still eliminate or reduce transmission. So they do a transmission experiment. Okay, so they give mice intranasally, intraperitoneally the PR8 and Sendai NP, or just PR8 alone, and then they infect them with Sendai luciferase on days 1, 3, 5, or 7 after the intranasal immunization. Okay, and then they put them in a cage with other mice, and they see if the other mice glow, because the virus has a bioluminescent marker in it. Right? So you can easily check that out. All right, so this is kind of interesting. When you co-house mice with naive contact mice 1 day after infection, transmission is observed in all groups, but at different levels. This is cool. 75% of contact mice, all the contact mice became infected when they're co-housed with an index mouse that got PR8 wild type, no NP from Sendai. 75% of the mice got infected fewer when they're housed with a mice immunized with PR8 Sendai NP intraperitoneally. And then finally, if you give them intranasally, the PR8 Sen NP, only 30%, 33% of the contact mice get infected.
I definitely want to be in cage 3.
Yeah.
Yes, cage 3. Yes, that's pretty cool.
Day 3 and day 7?
Day 1. That's all day 1.
Oh, no, they're day 1, of course. So we have more days to discuss.
Yep.
We have day 3.
Uh,
No transmission in contact when you give them the Sendai virus, the Sendai influenza peptide, PR8-SEN-NP. Let me be consistent. All right. So now if you wait 3 days, which presumably is you generating more T cells, right?
yeah.
Now you don't get any transmission, but there's still virus there. Okay. So you don't need to completely get rid of virus to stop transmission. So day 3, and then you get transmission in mice that either got intraperitoneally inoculated or the wild type influenza virus intranasally. Day 5, again, no transmission when you get, there's still no transmission with the intranasal PR8-SEN-NP, but now there's also no transmission with the intraperitoneally inoculated animals, so that by day 5, they have produced enough T cells in the lung, I guess, to take care of that.
The T cells finally made it there.
Yeah.
I know. It was
I mean, they also say that,
torturous trip.
but remember, the intraperitoneal inoculation makes normal levels of circulating TEMs,
Right,
right?
the effector memory. So some of them have finally made it to the lung.
Yeah, some of them.
Well, yeah. And what we're really looking at here is, I think, the length of infection in the vaccinated mice, right?
Mm-hmm.
So with the PR8-SEN-NP intranasally, the proper route, you get after day, well, by day 3 certainly, there's just no transmission happening, because if you look previously at how long the virus is actually in the lung, it's pretty much gone by day 3, right?
Yeah.
So, and then the others, the virus is still around for longer, and so you see it.
Day 5.
You see it transmitting.
So you still see virus at day 5, actually.
Yeah.
A little bit. But then it's a matter of amount. We don't know how much.
I can just hear the T cells, though, at that point. The swinging doors open. They shout out, "Boys, there's a new sheriff in town."
Why boys?
We are going to
Why
ride
not-
this metaphor all the way through the show.
Dixon,
No,
why not?
all the way into the sunset. Are you
Dixon.
Into
kidding?
the sunset. Yes, thank you.
Dixon, it's boys and girls.
Oh, is it
Not
boys
just
and girls? There's a new
Boys and
sheriff?
girls. The guy that gets the T cell stands up and says, "Boys and girls, we're going to do
I know,
this."
boys and girls. I see what you
Yeah.
Okay, boys and girls.
Now,
right.
okay, so basically what we found so far is that these TRMs in the lung can reduce the amount of virus made and shorten the window and also reduce transmission. Pretty cool.
Yeah.
And so the experimental setup is beautiful, right? Because you'd have no antibodies, you have no CD4s to help make antibodies. You just have these CD8s. It's
Yeah.
cool.
It is very good.
All right, so what's
In
the
this case,
mechanism?
it's not just CD8s.
Why not
It's
just?
CD8s.
You're right. You're absolutely right. You are not just. You are what you are.
Yes.
That's right. Thank God they came. I mean,
I'm
otherwise,
just a PhD student, people say. No, you are a PhD student.
that's right.
You are what you are. You're not just Dixon de Pommier.
No.
You are Dixon de Pommier.
I guess so.
We said that so long ago in the midst of the pandemic. People would write and say, I'm just a economist. I'm
Right.
just a plumber. I'm just a teacher. And we would always say, no, take out the just. All right,
Listen, I was on a trip. I have to tell you this story. Very
Okay.
brief.
I'll
I'm on a
take
trip
a
with
break.
my wife in Africa, and we're all telling each other what we did for a living to be able to afford this trip. And we get to this one guy, and he says, "I'm a plumber." I said, "Really, you're a plumber?" I said, "Where did you plumb?" He said, "Los Alamos." Not just a plumber. He worked on some incredible instruments, and he was the only one that could save the day, basically, in several cases.
Although, having hired plumbers, I would say that if you're a plumber pretty much anywhere, you could probably afford a safari to Africa.
This is all true. This is all true. But he sold himself so
Right.
timidly. He was
Mhm.
expecting your reaction. I'm sure he was.
All right, what's the mechanism of this protection?
Uh-huh.
Okay, so they took these peptide-specific TRMs and TEMs and did RNA-seq to see what's turned up. And they say, unsurprisingly, there's an upregulation of pro-inflammatory cytokine genes associated with cell-killing activity, and they list a bunch of those. And included among those are interferon gamma and perforin, which are induced in TRM cells, the ones in the lung, the tissue-resident memory cells, on day 3 post-challenge. And we know, though, those are important for T-cell function. So then they took knockout mice. This is what I love about this paper. They then say, we're going to take perforin knockout mice, and we're going to take interferon gamma receptor knockout mice.
So there's one other part of A and B that I think is important that we should mention.
Sure.
So here they're looking at different anatomic locations. So they look at bronchoalveolar lavage, lung, or spleen. And in the lung, they look at both effector memory and the resident memory cells. And you kind of assume the spleen would only have the effector memory cells, because why would it? They can't have the lung resident memory cells because it's not the lung. And they look at them sort of at day 30 after vaccination. So basically, at this point, the resident memory cells are just sitting in the corner, not responding to anything. So they have become resident memory cells, but they aren't actually responding to anything. And then they also look at day 30 resident memory cells 3 days after challenge. So it's sort of like, can those resident memory cells actually recognize that and do something early after seeing the re-challenge? And so they note that in the bronchoalveolar lavage and in the lung, they see at 3 days after challenge, these resident memory cells have now kind of woken up and are starting to do this and are making the inflammatory cytokines that Vincent mentions and have a particular gene expression profile by PCA. But the effector memory cells at day 30 post-challenge or day 3 post-challenge have not yet started to do something, whether they're in the lung, so they're there, or in the spleen. And so again, this is kind of saying the resident memory cells are the ones that are reacting really quickly in that first 3 days.
They're the ones that are quick on the draw.
Yeah, exactly.
There you go. That's right.
So they do a series of experiments similar to the ones we've described using these knockout mice. So let me just summarize them. So they do the co-housing, right? They do the immunization, they do the challenge with Sendai virus, and then they do co-housing. So to day 3 post-challenge, they get good transmission in the contact mice housed with the interferon gamma receptor knockout mice. Okay, so that's needed. Interferon gamma receptor is needed to reduce the transmission that they see, right? Perforin makes less of a difference. When perforin is important for cytotoxic T cell killing, so it's telling us now maybe that's not part of it, right? Not cytotoxic T cell killing of cells. So it's an interferon gamma receptor interaction that's critical for the mechanism by which the T resident memories are limiting the virus reproduction. So the next experiment is, they say, is these are all pretty close challenges to the immunization. So they want to know if we have pre-existing T resident memory cells, can they also work to protect the respiratory tract and limit transmission? So they immunized the mice, the contact mice now in the different ways to generate T cells. They can make TRMs or TEMs or just TEMs by intranasal or intraperitoneally. And then they look at Sendai transmission from index mice to contact mice. Remember, before they didn't do anything to the contact mice. They were just naive contact mice. Now they want to know if you immunize the contact mice with the peptide, can that protect you? And they wait, I don't know how many days, 35 days after immunization before they introduce someone into their cage. I'm having this of a classroom, right? Where there's
So-
one student in there and they're
Yeah.
immunized, and you wait 35 days. And
Wait
then they let the
35
other
days before a stranger comes to
But
town. Yeah.
before, in the previous figure, we were looking to see if I care, if my neighbor was vaccinated, if my
That's right.
neighbors being vaccinated protected me. Now I'm looking to see if me being vaccinated protects me from my neighbor.
So one student in a classroom all by themselves for 35 days does not constitute herd immunity.
No. That's what I was just. You needed 35 days to
No, I
make the
just,
immune response.
exactly.
Okay, so what happens? So I have everything. Basically, I have everything highlighted here. It's not useful, Vincent. The mice that were immunized with PR8 sen NP intranasally, they got big reductions in viral burden and the probability of infecting. Only 4 out of 15 mice got infected compared to 12 out of 13 when they just got the PR8 without the NP peptide, or the PR8 SNP peptide intraperitoneally. That didn't do very well either, as we saw in the donors before. So basically, if you're immunized and you're immunized intranasally with an NP peptide, that can help you not get infected.
Yep. So it's going to help you both not infect others and not get infected
Yeah.
yourself.
And they do the same experiment in knockout mice, and they show the same mechanisms are at play. In other words, interferon gamma is really important, and perforin doesn't matter all that much. And interferon gamma signaling, so it's a big deal here. So what does interferon gamma do? So they said, well, maybe this results in recruitment of, so that maybe the TRM recruit innate immune cells into tissues. But they say, well, we looked and we don't see any more NK cells or monocytes recruited in on day 2. So maybe that's not what's going on here. So then they said, let's look at what kinds of cytokines and chemokines are being made. And these mice that get the peptide have a distinct proteome that is made compared to, say, mice that don't get a peptide or that get the peptide intraperitoneally. They have upregulation of genes, not just general inflammatory genes, but genes associated with antiviral responses and antigen presentation, basically.
Yeah, and this isn't just in the immune cells. This is actually in the airway epithelial cells. And so the presence of perhaps interferon gamma is changing other parts of the lung to make the lung better at presenting antigen.
Right. So, and then if you knock out interferon gamma receptor or interferon gamma gene, you do get inflammatory responses, but you don't get this increase in antiviral immune-associated genes. And so that's what the TRM cells are doing. So it's via interferon gamma. They're making interferon gamma, and that gamma is somehow turning on an antiviral program in the lung.
Is this information useful in understanding something about the cytokine storm in SARS-CoV-2? He asked innocently.
I hadn't thought about that, so I'm trying to think it through now, and I don't know.
Okay.
Maybe.
That's a good answer. Let's find out.
I do, but where I thought you were going is slightly different. Does this help us understand more about the role of T resident memory? And I think it does, right?
Oh, I'm sure of that.
I think it
would
tells
agree
us
with
we
you
should
there.
be trying to get those immunized in our vaccines, right?
Yeah, well, I think it also tells us, because normally we would think about the point of having a resident memory T cell is to kill the infected cell.
Right.
And it's not just about killing the infected cell. It's actually about changing the immune environment as quickly as you possibly can.
Right.
And so it's not about, can this cell kill the virus infected cell? It's, can this cell turn on other immune responses by its early quick presence and early quick action.
Yeah.
Uh-huh.
So
And
it doesn't
so-
really shoot; it organizes a posse.
Yeah, but
There
we
you
can-
go.
no, it's exactly right. And we should think about, we shouldn't be trying to imagine strategies to make things shoot. We should be trying to come up with strategies to get that posse organization
Just to
happen.
put them back in jail would be fine.
Yeah.
So then the next question is this durable? Does it last?
Uh-huh.
So they wait 6 months after immunizing with the peptide, with the
Which is
T-cell
a very long time in a mouse life.
It's a quarter of its lifespan, right?
Right.
Absolutely.
Let's see, 6 months after immunization, contact mice immunized with the influenza, with the Sendai. Oh, where did I see this? Wait, where is it?
You should have highlighted it.
Hey, you're making fun of my highlighting. All right.
You're
let me just
using
tell you the
fluorescent
results.
green to highlight with.
Okay, that's a separate experiment that I was just thinking about. Okay, so this one, 6 months after immunization, the contact mice that were immunized with the Sendai peptide have no protection against infection compared with control mice immunized with just the wild type virus. But they did have much less virus in them. So 6 months after, you still get infected, but from a mouse put in the same cage by transmission, but they make less virus, right?
So while we cannot ask the mice how they are feeling,
That's right.
We may be seeing a situation of protection from more severe disease, even if we don't see protection from infection.
Right.
Yep.
Yes, and also with lower viral titers, they may be less likely to spread.
Yeah. Is there weight loss with this?
They
'Cause
don't.
that would be an
mention that,
easy
no.
parameter to
They don't
measure
mention it.
without having to.
Yeah. It's a common one, but they don't do that. No. So 1 month. So that was 6 months, but 1 month post immunization, there is protection. Now look at this. They use a different vector to deliver the nucleoprotein. They use an adenovirus vector. 6 months, they get really good protection against transmission. And they get TRM cells in the lung. And so you just change the vector and you get a different result. It's very interesting. So you cannot assume that just giving the peptide is going to be the same no matter what, right?
You
This
can't
is not a
assume that your
replicating
vector is
adenovirus,
entirely
though, is it?
neutral in the process.
Just the
That's right.
adenovirus
That's
does not replicate. Is that correct?
It's a non-replicating adenovirus. Yeah.
Right, so
It just
it's just
encodes the
different.
Sendai nucleoprotein peptide, but they get 6
Fascinating.
months. It's durable for 6 months. Very cool. So then they put all their data together and they see there's a negative correlation between total viral burden and the number of TRM cells in the bronchoalveolar lavage, the lung, and the nasal cavity, with the strongest correlation in the nasal cavity. Right? Not surprising, right? That's where the virus
where you
comes
put it.
in.
Yeah.
Interesting. And a
How
negative
many T cells
correlation.
can you get out of a mouse nose?
Good question. Do you know how many brain cells there are in an ant?
I don't.
I had to look it up the other day. 250,000.
It's
Just
Sizable
and there's a
number.
worm that goes in there, Dixon.
yes, there is.
Also, negative correlation between the number of TRM cells in the same place and probability of infection. So the other was probability. It was total viral burden. This is probability of infection. In this case, the correlation is in the lung. It makes sense, right? So probability of infection getting established, it's in the lung, and that's where the protection is. But just viral burden is in the upper tract.
The protection from infections in the nose, the
Yeah,
protection from severe diseases in the lung.
that's right.
Right.
That's right.
Now, alveolar
Okay.
macrophages play no role in this whatsoever. Is that right?
No, they looked and there weren't differences in the vaccine groups, and macrophages were NK cells. Yeah, no role in this.
Right.
All right, so this is what I was thinking of before. So they say, we have some vaccines, we have 2 doses, there's a prime and a boost, and maybe the boost never gives rise to T cells because there's a the antigen's cleared because you already have some kind of memory, right? So let's look at this. Does pre-existing TRM-mediated does pre-existing immunity limit TRM-mediated protection against infection? All right, so mice are immunized with either wild-type influenza virus or mock immunized, and then they're given influenza virus sendai NP or adenovirus sendai NP. So that's like a prime boost, but it's a heterologous prime boost, right? The first is, they're two different virus vectors carrying the NP. Okay. The mice that got pre-existing influenza virus immunity were not protected against sendai virus infection after they got the peptide. Remember, in the previous experiments, they had much reduced virus loads and transmission and so forth. But adenovirus, on the other hand, gave them protection mediated by TRM. So using a different virus for the boost makes a big difference. And the idea would be that if you use influenza virus twice, the second time is being cleared before it can do anything, right?
Right. But it's because of all of those evil antibodies and CD4 cells being a problem.
Evil antibodies.
That's right.
Now people can realize I'm a T-cell immunologist.
So they also asked, does this heterologous prime boosting affect the longevity of protection? Remember, the influenza only gave us 1 month of protection. So they did this same experiment. And again, if you do the heterologous prime boost, you get 3 months of protection. They didn't look so far out. So basically, the number of virus-specific TRM cells in the respiratory tract dictate the ability of cell-mediated immunity to protect against, well, I wouldn't say protect against infection. I would say to modulate infection, because it's not sterilizing, as they say.
No, right.
They say later, they say these protective immunity provided by these CD8 TRMs is not sterilizing, as the cells must first encounter their antigen presented by a virus-infected cell. So it's not sterilizing. It's a virus-infected cell, right? And I don't think you can ever make a sterilizing vaccine, but that's another story. So that's the story here.
But you did prevent transmission.
Oh,
Yes.
Here's the other thing. They reduced it. Although at some points they prevented transmission also.
Yeah.
Yes.
Yeah, that's
like
right.
day
That's
3 and 5 with the
Exactly.
Influenza NP. Yeah, they did prevent transmission. But they say, now we're not going to use one peptide in humans because the MHC is diverse in the so one peptide is not going to suffice for all. One peptide cannot rule them all, right?
No.
So they said, what the hell? Just put the whole protein in, put the whole nuclear protein in. And while you're at it, put in other proteins that don't give rise to neutralizing antibodies. So this is a proof of concept here with using a single peptide, because the mice are all inbred and they all have the same MHC, right? Let's see, what else? Oh, so the other question I think is very interesting. So they say, an interesting question is, what's the importance of TRMs in different locations in the respiratory tract? And you say, we didn't address upper versus lower, but probably the upper tract TRMs have a bigger role, right? Because they're right there at the portal of entry. So that's where you want to get them. And again, remember, the CD8 TRMs, they respond to infection. They make interferon gamma, and that makes the respiratory epithelium turn on an antiviral program. And they say maybe you don't even need contact of the TRM with the epithelial cells. I guess not, because, one TRM cannot contact so many epithelial cells, right? But a diffusible protein could.
Well, and I think that that's a really interesting thing to really be thinking about, is if the goal is to get a diffusible protein that's inducing this antiviral response, that might be something you could do in a different way than if you have to get a T cell to be able to contact the virally infected cell.
And I just want to end by reminding everyone there's no antiviral antibody in any of these experiments. Right? No antibodies were harmed in these
No.
in this work.
No.
So this, please believe in T cells. You have to believe in them. Although science is not a belief system, so we have
No.
data here that shows
Yeah.
that,
That's right. That's right. You have to believe the data.
So you have to make vaccines. Well, it probably would be good to make vaccines that make both antibodies and
Yes.
T cells,
Yes.
right? Not just one. But more evidence for T cells. I like that. Really cool stuff.
Yeah.
Yeah, no, it's really good.
And the T cell blows the smoke off its pseudopod and sits back down to its drink.
Exactly right.
You got the voice
That's
too.
right. That's right. That's right.
My. I just want to read one email because it is a passing. Right? Does that sound like a plan?
That
Sure,
sounds like a
sure.
plan.
Sure.
Alan, can you read that one from
Yes, this is from Steve, right?
Yeah.
Dear Microbe, I thought you might be interested in a notable passing. Steve sends this from Wolfson College. In memoriam Sir Anthony Epstein. It is with great sadness that the college has been informed of the death of Sir Anthony Epstein, 1921 to 2024, who died after a very short illness in the early hours of Tuesday morning, 6 February 2024, at the age of 102. Sir Anthony was an extraordinary fellow, 1986 to 2001, and later honorary fellow, 2001 to 2024, and his obituary is expected to appear in the Times the next few days. The college understands that there will be a small family funeral, perhaps followed by a memorial event for which the details are not yet known. Further details, including arrangements for any messages of condolence, will follow in due course. And that's from Luke Jackson Ross, the college secretary at Wolfson College, which is at the University of Oxford.
And yes, Epstein-Barr virus.
Yes. That Epstein.
Yeah.
That Epstein. 102.
102.
Sir Epstein. Sir Anthony.
Sir Anthony Epstein, yes.
And indeed, he was an extraordinary fellow, whether he was an extraordinary fellow or not.
Yeah, I caught that when Alan was reading as well. I think he was probably an extraordinary fellow for more than just 1986.
For more than just that period of
2001.
time. I think you don't stop being extraordinary at the
You don't.
when you become an honorary. Yeah.
You know any
And
other?
I don't think that's the same as the League of Extraordinary Gentlemen. Although it's
No,
possible that he had
no.
superpowers, we can't exclude that.
We didn't do
But
the
what-
experiment.
Yeah.
Um,
A giant in the field.
Does anyone else know who has been given knighthood or damehood as a scientist?
Anybody?
Stephen Hawking?
Yeah, he was.
He was?
I think so.
I think Darwin was.
Let's see.
Okay.
I don't see. I don't recognize names here. That's funny.
So I just Googled Order of the British Empire scientists,
Yeah.
and I get Stephen Hawking, John Bell, and Jane Haley.
Okay. I have a big list
actually,
here.
I've got a longer list. Isaac Newton,
Yeah.
Yes.
Tim
Right, goes back a
Berners-Lee,
way.
Sir, yes.
Jane Goodall, Colin Blakemore, Sarah
Maurice
Gilbert,
Wilkins. Maurice
Andrew
Wilkins.
Pollard.
Maurice Wilkins.
Sir Alec Jeffreys.
Darwin, not?
Stephen Hawking, right?
Medawar,
Peter
Peter
Medawar,
Medawar.
right?
Matter of-
Also, I saw Tim
Alexander
Berners,
Fleming.
Tim Berners-Lee.
Yep.
Goodness.
You got to do big stuff to get that.
You do.
You get knighted or
Yes.
damed.
Paul Nurse.
Paul
Nurse.
Nurse. Hmm.
And then you can be called Sir or what?
Oh,
Dame, right?
Dame.
Elton
Dame.
John.
Paul McCartney, right?
Right?
Yeah.
Yes.
Yeah, that's right.
Terry Pratchett.
Keep going.
Anyway, Sir Anthony Epstein, 102, not bad.
No, that's
I don't think you can complain about that, Dixon. right?
No, that's
not.
a good one.
No, I wouldn't have. I would have a smile on my face.
I think you're going to make it to 102, Dixon.
I hope so. I would like to. I'd like to know who I was at 102. Because if I don't want to live to 102.
You're
I wanna live
Dixon de
until I-
Comier.
Well,
Yes.
I know, but
TWiV 2000.
TWiV,
Hold on, I
or
got
bust.
somebody knocking at the door. I'll be right back.
Uh-oh. So the title of this show, Alan, I think we should come up with something like Blazing Sendai.
T cells at the OK Corral.
That's right. That listen. You know, when you leave Dodge, in this case, you're actually dodging something. Get out of town, right? No, I think a lot of good analogies, Western analogies would be
It's
good.
just the Amazon guy. I don't need to get up to get
The
that. You know?
Amazon guy.
No.
Why do they
Prime
knock?
or regular?
What?
Prime or regular?
I don't know. It's just well, we have a Prime account here at the incubator. Yeah,
All right.
saves shipping.
Right.
And we order so much stuff that
It
pays.
Indeed.
Like
All right. Back to the show.
stuff we need.
Indeed.
All right. So we read that email. Let's do some picks, Dixon. You're up first.
Yes, sir. Okay, so I've been neglected with regards to my interest in the crossover between science and art and science. And we've all agreed in the past that there is a tremendous crossover. Without being able to envision data in a way that your optic lobes can process it, a table of numbers leaves me cold. But a picture of that table of numbers that you can actually visualize is something of beauty that is rare. So I have located this website by Scientific American that actually has a very extensive list of ways in which art has aided science in visualizing information. And it's really a wonderful thing to look through because you look through it. You don't have to read it necessarily, although you probably do eventually, but just to look to see the way data is presented, you can see the history of the development of, and then, you know, the circular charts showing how all life on Earth is related. And, you know, the more you expand that, of course, the more you see. There had to be a first person that could do that. And my first person pick, although we could start a contest to see who's the very first genius at expressing scientific data in an artistic way that lets everybody get it. There's no way to miss it because when you see it, you believe it. And so that's why I say seeing is believing. Ernst Haeckel was an amazing science illustrator and a scientist. And if you didn't know that, and I know that everybody listening here at the podcast did, but if you listening out there had never experienced an Ernst Haeckel poster, just go on Google Images and type his name out and illustrations next to it, and you will be astounded at the beauty and the intricacy and the amount of time it took to just create those wonderful pictures of real life objects. And they're elevated to something else because they're not just depicted, they are arranged. They fit together like a puzzle. And he envisioned this puzzle prior to making the drawing. So he had to spend hours drafting and getting examples of the same specimen to make sure he had all the parts spoken for. And it's a very difficult chosen lifestyle to develop into a very picky, I would say, medical illustrator that refuses to leave anything out of the picture that isn't there, or refuses to put something in that's not there. So Hegel was the amazing first in my book. I don't know who preceded him. I have no data on that whatsoever. I could say that Leonardo da Vinci was a wonderful illustrator, but he actually drew the heart wrong. And I learned that from a good friend of mine, a medical illustrator, who said, although Da Vinci was a fabulous draftsman, he was not such a great anatomical person, and neither was anybody else for that matter. So he was like one of the first, right, for Vesalius. So I don't know. I'm just so struck by the absolute beauty, intricacy, and detail of Haeckel's drawings and
Cool.
charts. So that's my pick of the week.
It's pretty cool. I like how they just put the animals first, and then they start to fill in the chart. Right?
It's
see how it's
really
built.
remarkable,
It's
actually.
very cool. Nice. And also, I noticed on the sidebar, some other articles, and there's one about these obelisks that have been
Yes.
found. And that's
Yeah.
a preprint. I'm waiting for it to be reviewed, and then we'll do it.
Good, because I'm excited to think and talk
That
Yeah.
should be fun.
and have a conversation about it.
Good.
Brienne, what do you have for us?
Last week I was part of a film fest where some science related documentary was shown, and then we were talking about it as experts in the field. In fact, the director was also on the panel, so I got a chance to meet him too. And the documentary that was shown is called Vaccination from the Misinformation Virus. And so it's really about reasons why people are vaccine hesitant or are not getting vaccines, and kind of talking about why vaccines are important. It involves interviews with a lot of scientists, as well as pharmacy students, pharmacists, and sort of people kind of throughout parts of science. And it's really sort of meant to be informing a lot of people. If you go on the website that I've linked and you click on the PBS part at the top, you can actually watch a full version of the documentary there. But there are also a place where he lists media resources. And one of the resources he's made are, I think he called it link outs or lift outs. I think it was lift outs, which are small video captures of different pieces of the documentary that are meant to be shared on social media. So you can have a short clip of Betty Korber talking about the importance of vaccines that you could share out, or things like that. And so I thought that was a really neat approach. And it's a great documentary for people who are interested in thinking about reasons people aren't getting vaccinated. That's the one that I watched and discussed, but he has another documentary that has come out much more recently called The Invisible Corpse, which is about the public health infrastructure and public health workers in America. So I thought both of those might be of interest to our listeners.
Is that corpse or core?
Core.
It might be core.
Core. It's core.
I said the invisible corpse. That sounds like a
No, they didn't
an
find.
H.G.
They
Wells
didn't finally
movie.
find the body. It's
Yeah.
the invisible core. It's about the public health service.
I'm sorry, I
That's
didn't.
really good.
I
No,
was
it's
just.
totally fine.
Thank you, Brienne. Alan, what do you have for us?
Well, we didn't plan this, but mine is slightly linked to Brienne's pick. So this is it's just an NPR article, but the thing that it links to, I thought, is a really cool effort to try to reduce or eliminate the pain and trauma associated with childhood vaccination, and try and prevent needle phobia, nip it in the bud. The idea is, there is a significant fraction of the population that is terrified of needles. And I have to say that I'm not a huge fan of them. I don't know if I quite have needle phobia, but I don't look when I'm getting my flu shot. And this stems, of course, from one's earliest childhood experiences, which may include being held down by a doctor and getting a painful shot in your arm.
Mm-hmm.
This also is traumatic for the parents, I can now say with authority. When you're holding your child and they're getting a painful shot in their arm and they're looking at you like, why are you doing this to me? That is a pretty terrible experience. And you can't explain to a 1-year-old what this is doing. So this is an effort led by a pediatrician, what is the guy's name? Stefan Friedrichsdorf, who's at UCSF. And he has developed a protocol for dealing with this, for preventing this kind of traumatic scene in the pediatrician's office. It's basically a 3-step thing. You apply a numbing cream to where the shot's going to be delivered.
Sure.
And then the child is held ideally on the lap of the parent, so they feel safe. And the third part is that they're distracted. You give them a lollipop or a little toy or something, or if they're an infant, they could nurse or they could suck on a pacifier. And in doing this, the kid is preoccupied and feels comfortable, and the shot goes in and they don't know it.
Right.
And then you have one less person who's needle phobic.
Exactly.
So I thought this was a great idea and a great effort.
That's awesome.
Yeah, and then they see the needle, then they start to cry.
Well,
That's
no,
after
you don't
it's
let
been
them see the needle.
withdrawn.
And
After
I
it's
have
been
to say, I
withdrawn.
think veterinarians may actually be a step ahead on this.
Yeah.
When I took Theo, our newest cat, to the vet for his initial visit, and he needed one more shot. They hadn't given him all of his shots, just because of sequencing. He hadn't quite gotten all of his shots at the shelter. So there was one more he had to get, and he's a great little cat. He's very friendly, and the technician held a squeeze treat, which is a tube of basically cat pate. I think it's utterly disgusting, but cats are
Love it.
mad for it. So the technician held the squeeze treat open, and Theo is there like, "Ooh, squeeze treat, this is the best. I love squeeze treats." The vet comes in with the needle and shot, and oh, yummy squeeze treat. The cat had no idea he'd been vaccinated.
Right.
Cool.
And not that kids are animals, but well, they kind of are.
They still like treats.
Yeah, they still like treats.
Alan, do you mind having an IV put in?
I don't like any kind of I don't like
I watch
injections.
when they draw blood. I watch.
No,
Because I want to see how well
when
they do.
When I give blood, it's like, okay, I'm looking over here. Yeah.
Nope.
I like to watch. I watched recently, and I remember she went in, and then she pushed it all like an inch in,
Mm-hmm.
and I don't have a problem with that. I think it's cool. I like to talk to them about it. I say, "Hey, I have really good veins, by the way. Look here." They, "Oh yeah, you do." And then on your wrist, if you have visible veins, you can show where the valves are by pushing down and then moving your finger forward, and it remains collapsed to the valve. And then if you lift, then the blood flow resumes. It's very cool. And I've done that with a number of venipuncturists, and they're like, "Yeah, look at mine. I'll show you my valves. Let's compare valves." All right. It's not that bad. It's okay. You guys don't like it. Okay. Thank you, Alan. I asked Alan this week for that website that shows nice pictures of kids getting vaccinated because I wanted a picture of a kid getting vaccinated for Paul Offit's latest video, and he gave it to me. And I think I should tell people now what it is, since
Isn't it from it's from a magazine?
Yeah, Self magazine got together
Yeah.
with is it American Academy of Pediatrics or somebody, and because this is pre-pandemic, the editors at Self,
Yeah.
I guess, needed a stock photo of somebody getting a vaccine, and all the available ones were horrible. And so they said, okay, let's shoot some. And they shot a whole gallery, and they're free for download.
Right. Okay.
But that's not your pick.
That's not my pick. My pick are two new DJs that I've discovered. Now, about 2 years ago, during the pandemic, I got interested in electronic dance music, EDM, and I picked a couple of DJs. Two DJs. I picked Chris Luno and Miss Monique. And I've since been listening all the time. We play music here at the incubator. We actually bought some wireless speakers, and we play music. And mostly it's what I pick. So I've been playing EDM, and it's on YouTube. And you go there, you play, and then they'll recommend new people. So I'm meeting new DJs all the time. And I have two which I think are fabulous. So one of them is Rosana Nun, who is from Spain, and has just 41 videos on YouTube, but really very, really interesting and different kinds of DJ. And also she often does vinyl. She'll have two vinyl turntables, right? And, mixes between them. And it's very interesting because she'll have one playing, and then she's looking through her vinyl collection, through the boxes she has of all these LPs, and she pulls one out and she listens, and she goes, "No." I don't know how they do that, because nowadays you just get electronic libraries, but she uses vinyl. And it's very funny because you can hear the crackling sometimes, right? Because they're scratched. So I really like her. And she doesn't always do vinyl, but the thing about DJs now on YouTube is that they go to different places and record in different venues, like outside or next to a beach or in a park or something. This is the latest thing, right? In the winter, in the middle of a snowstorm, they always do different stuff and try and outdo each other. But she's mostly in her home, I think, where she's got her record collection. I like her a lot. Rosanna Nun. And then the other one is called Sunset Cartel. This is a collection of DJs. And they're all different. They're men and women and different styles as well. But they're all very, very cool music. Very different from your usual electronic music. So I think you should check them out if you like EDM. I think it's really cool.
Nice.
Little non-science pick.
Yeah.
We have a listener pick from Mary. Hi, I've been a dedicated listener watcher since I discovered Microbe TV. What a great educational and humanitarian service you provide. I'm watching your virology course episodes, and although I have a degree in biology, it was more than 40 years ago. I'm learning a lot. I have a listener pick from the early 70s for TWiV, if you should happen to enjoy it. It's a film called Protein Synthesis, an epic on the cellular level, and it's a super fun happening from the early 70s to illustrate what was known about protein synthesis at the time, introduced by Dr. Paul Berg. You can see a young Paul Berg at the beginning.
tRNA.
It's great. We've had this
Yes.
before, but it's just wonderful for new people who haven't seen it. Go check it out. It's just a lot of fun. All right. Speaking of a lot of fun, the fun is now over, folks. That's TWiV 1089. You can find show notes at microbe.tv/TWiV. You can send us questions, comments, pics to twiv@microbe.tv. And if you like our work here, it's not really work to us. We just love it. It's our passion. You can help us, support us, please, financially. Microbe.tv/contribute. Dixon, Despommier, Trichinella.org, thelivingriver.org. Thank you, Dixon.
A good comic romp through the world of viruses. I enjoy
And immunology.
And immunology, excuse
Don't
me.
forget,
Immunology also.
you know, they are inextricably entwined.
They are.
You cannot really separate the two.
No, nor should we try.
Alan Dove is at turbidplaque.com and alandove.com. Thank you, Alan.
Thank you. It's always a pleasure.
Brian Barker's at Drew University. Bioprof Barker on Blue Sky. Thank you, Brianne.
Thank you. I appreciate the love for immunology and the fact that the two fields are so intertwined, and I learned a lot.
Nobody twisted my arm. I just learned. This is the way it is. I have to say, the best way to understand something is to study it. And that's what we do here on TWiV, on Immune. I've been having a long-term course on these subjects, and I really, you begin to appreciate how they're related. Not just how they work, but how they're related.
I have to add my 2 cents on this also. I'm an old school person, and I was a good friend of Elvin Cabot's, who was a leader
You
in
were? You were?
Cabot. No, I'm a very good friend of Elvin's. But Brianne, you make this understandable. And
Thank you.
that's the virtue of being a great teacher, and you are a great teacher.
Thank you.
And you can see Brianne's lectures on YouTube.
You
Just
can.
search Brianne
I
Barker.
can't, why would I do that when I can have them right here?
Well, what if you really wanted to learn all the details of some part of immunology?
Well, you know.
Yeah, you should do that. Or you
I'm
could
with
do
Paul Berg.
You could do that. You could take virology. I have a virology course there. You should teach a parasitism course.
Well, I do have a parasitology course. It's online with Parasites Without Borders, and you can have
It's
5
TWIP. The first 20 episodes of TWIP, right?
and first 20 episodes of TWIP, and so two ways you can get.
Oh, so there's
lectures
your lectures are on Parasites Without Borders?
all of them. You recorded them.
Oh. No.
No, I
Oh,
put
that's
you to
right.
sleep. I remember I put you to
Oh,
sleep.
gosh, I remember. We did it in my office. Put up a green screen behind you.
We
Yes,
did
yes.
that, and we did
And I
that.
did doze off at times. You're right. I totally admit it. That was a long time ago. I was younger.
He learns better when he's asleep.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV. is viral.